Hey, this is Mr. YS, and this is a clip on post-transcriptional modification and gene expression. So the first question is, what is post-transcriptional modification? We've already looked at the process of transcription. Transcription was taking DNA, which is the double-stranded molecule of inheritance, and then from it, using RNA polymerase, making a single-stranded mRNA. And so what we're talking about is post-transcriptional. We've made the transcript. So now what happens afterwards? And you can see there's some modifications, some changes that are going to be made. So what kind of cells does it happen in? Well, the type of cells, we're going to have a look at a picture in a second, but the type of cells that it happens in, they are eukaryotic. And how can it lead to regulation of gene expression? We'll get onto that in a minute. Here are two types of cells. We have eukaryotic cells, which are characterized by having membrane-bound organelles. So we've got the nucleus, which is bound in a membrane. We've got other things as well which are bound in membranes, other structures like mitochondria. But eukaryotic cells contain their genetic information inside. So it's membrane-bound. And that means that transcription and the processing that happens of it occur in the nucleus. So we've got these two bits, transcription and post-transcriptional processing. Both occur in the nucleus because they have a nucleus. So over here we can see a bacterium, a prokaryote doesn't have a nucleus. So that means all of transcription and, well, there isn't any post-transcriptional modification, but then the next stage, translation, it all happens in the cytoplasm. But in eukaryotes there are two locations, nucleus for transcription, nucleus for post-transcriptional modification. And then next we'll look at the process of translation, which takes place outside in the cytoplasm. The first thing that happens post-transcription is we can see two things here. And you can see we've got 5' up at this end and 3', and that's talking about the direction of the RNA strand. And so you can see we've got this G. So this is a 5' cap. We'll have a look at what type of cap it is, because it's a G, it gives you a clue. And then down at the 3', you can see we've got A. So we've got the nucleotide, we've got a poly means many, we've got a many A tail, a poly A, 3' tail. Now don't get overwhelmed by this, we don't need to draw this structure, it's just to show you what it is. So the first thing is, you have a 5', and we can summarize it as a methyl cap. 5' methyl cap. So why does it have this? Why does it have a cap? Why do anything afterwards? Well, the cap protects it from enzyme breakdown. So it means that when you put the cap on it, it won't get broken down by enzymes, which would normally degrade RNAs. So this cap is a protective cap, and the same thing is true of the poly A tail. So what kind of a cap is it? Well, if we followed the mRNA along, so this is the mRNA, at the 5' end, we've got our sugar phosphate backbone of that chain. So you can see the phosphate here, then we've got the sugar, and then the cap bit is 1, 2, 3 phosphates, and it is using the nitrogenous base guanine. So it's guanosine. And guanosine is when you've got these 2 bits. So that's basically what it is. It's a cap using guanine as a base on the end, and we call it a 5' methyl cap, and that's going to prevent enzyme breakdown. And part 2 of our modification post-transcription was to add in this poly A tail. So adding in 1 to 200 of these adenines. So it's not just a few, it's hundreds stretching off this way. And that also is a signal for it not to be broken down by enzymes. So following transcription, we now got to the point where we've got a 5' methyl cap and a 3' poly A tail. And that's not the end of it. So once we've done that, now there needs to be some chopping and sticking together. Looking here, you can see we've got a code, and this is going to be an analogy for what happens in our next step. So the next step is called splicing. Splicing means to cut and stick. So we're cutting and sticking together the RNA, the mRNA. Okay, so look at this and try and read out by looking at that and see if you can see a code. You can see we've got these sections which are highlighted here. First one, AMAPQ, well that doesn't mean anything, and ZAPTQM. So I want you to try and cut these out and stick the message back together and see if you can work out some sense in it, because there is sense, or there is meaning in the mRNA, but in between it are bits that we have to cut out because they don't have a meaning. They don't lead to amino acids being made. So try and work out what the code says. If we cut out the purple and stick together the remaining, we can see that if we're talking 3-letter codes, which is what the genetic code is, we've now got something that makes sense. We have cut out bits that didn't make sense, and now we've got a message that we can read. And this is the idea of splicing. To cut out parts of the RNA to make, in the end, what we call a mature mRNA. If we look here, we've got some sections there which are going to be cut out. And we've got then the sticking together of the RNA to make the mature. The regions which we want, the ones which are in the kind of the red and the kind of faded red, they're called exons. So exons they're containing the genetic code for amino acids. They code for amino acids. Whereas these other regions which we need to cut out, those are introns. And so these do not code for amino acids. And so in the process of keeping the exons and removing the introns, we've now made our mature mRNA, which has the meaning, it has all the information to code for a polypeptide. All along it will have sections of 3 and 3 and 3 bases. UAG being the first, GGG and so on, and each of those is going to give us the code for one amino acid. We'll get to that next in translation. The next question is then, well, how do we do the chopping? What is the thing that does the cutting and the sticking together? And you can see they look like beans over here, but we've got some things which are coming together to create a loop in the mRNA and to chop out the regions that we didn't want and stick together the rest. First is a little review. Can you work out what end this is and what end this is? What we would call these regions. If you can see at the end, this is what's produced. So just stop and see if you can recap that. Okay, well, this end with the methylguanosine, this is the 5' methyl cap. And this is the 3' poly A tail. And then this region here, it's the thing that's being kept at the end, so that's the exon. And this bit here is being looped and held by these beans, the bean-like things, to be removed. So that's the intron. Okay, so what are these things? These things are called SNRMPs, SNRPs. And these are small nuclear, you don't really need to know this, but if you're wondering what it stands for, small nuclear ribonucleic, because it's got RNA in, ribonucleic protein complexes. So it's got protein and RNA. And so that's made in the nucleus, and then they form with amino acids, with proteins, they form this complex. And the whole thing is called the spliceosome. Spliceosome. The spliceosome. So the spliceosome is a complex made out of ribonucleic acids and protein. And what it does is it holds the mRNA and it cuts out the introns. And this is going to be, so this is called excised. If you cut something out, it's to excise it. So its whole job is to take what's called the pre-mRNA. So at this point, it's not yet mature. It's the pre-mRNA, chop out the introns, and then in the end create the mature mRNA. And here's our summary recap. So post-transcriptional modification is modification of pre-mRNA which was formed from transcription. It happens in the nucleus, and because it happens in the nucleus, it can only happen in eukaryotic cells. It involves the process of splicing, which is done by the spliceosome, which is an enzyme complex made of SNRPs, which removes introns, which are non-coding regions. They don't code for amino acids, and sticks together the exons, the coding regions of the mRNA, to produce the mature mRNA. Also, and this really is before, we have, so post-transcription, we have a 5' methyl cap, and we have a 3' poly A tail, which protect the mRNA from digestive enzymes. And this is also a source of regulation. So we can regulate the way in which things are spliced and the way in which we make, so it can be regulated, we can change it. We can increase or decrease the formation of mature mRNA by regulating splicing. And we can also, as we'll see next, we can make different things through splicing in a different way. Our next question is, what is alternative splicing? And how can it be used to bring about different functions for proteins? Let's have a look at this picture here. And again, don't get overwhelmed. There's a lot of stuff on this picture, but let's just follow it through. So we start with DNA, which has regions that are coding. And in between them, so these are coding for amino acids. But in between them are regions that are non-coding. These are our introns. And these can be removed. So the first way that we could remove these, if you look at the numbers here, we've got 1, 2, 3, 4, 5 coding regions of the DNA leading to the same 5 coding regions of the mRNA. And then the next bit is where the spliceosome is going to chop out the introns. Now what it can do is it can chop out different regions. It can chop out different introns, or in fact not include the exon. So if we look at the first thing is we can just get rid of all the introns and stick all of the exons together. And now we've got a mature mRNA which has exons 1 to 5. And that's going to lead to a particular protein, particular polypeptide, which folds up in a particular way. But if we look at what's happened in option number 2, well, in option number 2, you'll see that it's missing exon number 3 was not included. And that means that this is now a shorter polypeptide and it's missing this region, which is going to give it a slightly different function. And over here you can see that it's missing region 4. Region 4 was chopped out of this one. So it's missing, you can see that bit here, this fold is missing over there. So using the same gene, this is one gene, by chopping out different, splicing together different exons, we can end up with different versions of the protein or the polypeptide. So what's an example of this? And this is, I think, fascinating, that we've got this muscle protein that we'll look at in muscle contraction, which is called tropomyosin. And the gene for it has 11 different exons. So by splicing it together, we can make 5 different forms of this protein in our muscle. So we have skeletal muscle. This is the muscle that we consciously use and contract. And that's made by chopping out exon 2. But in smooth muscle, the muscle in our digestive system, we make that completely different type of muscle by chopping out exons 3 and 10. We leave in 2 and we put in 3 and 10. And so from the same gene, from the same DNA, from the same pre-mRNA, we can have different proteins made which then have different functions. Here's our recap. Alternative splicing is a process where different exons can be spliced together. We remove the introns always, but which exons you glue together means that we can form different forms of the mRNA, leading to different proteins being formed, e.g. tropomyosin in our muscle tissues, where we have 5 different forms due to alternative splicing. And that leads to skeletal muscle being different from smooth muscle. Our next question is, how is gene expression regulated? And what we mean by gene expression is we mean transcription of the gene, then leading to the formation of protein. And that's in our next step, that's translation. But expression is another way of saying kind of the use of the gene. And the way we use it is by using an RNA polymerase to transcribe it so that we've got then a copy, a transcript of the message. So how do we regulate this first step? So if this is step 1 in making a protein, is to transcribe it, and this is step 2, then how do we regulate the first step? Because if we increase this, then we're going to increase this. It's like a production line. If you can increase the first part, that's going to then increase the total output. We could decrease as well. So gene expression is something that can either increase or decrease, because it's the transcription of a gene. It's the we could think of it as the amount of mRNA which is being produced. And then that amount of mRNA will then influence the amount of protein in the end that we make. To explain this, how we can have regulation of expression, let's think of an example. So here's E. coli, which is a prokaryote. It's a bacteria. Now it wants to conserve energy. And you do that by not making the things that you don't need. There's some things that you need all the time, but there's some things that you don't need. And so E. coli, what it does, it's able to create enzymes that it can use to put outside. Because it's food sources, it's a bacteria, it will grow on food. It can then digest food outside of it. And then it can absorb those digested foods. And so it wants to respond to the environment it's in. If it's in an environment where there is a particular food source, it's going to start making the enzymes for that. So let's imagine the example of lactose. Lactose is the milk sugar. So you get E. coli in milk. So if you've got a glass of milk and you leave it for long enough, you'll get bacteria multiplying in there and they'll break it down and it will go sour. And so these bacteria are digesting, amongst other things, they're going to digest the lactose. So let's imagine there is no lactose around. Your E. coli is not in milk, it's not around any lactose. If that's the case, it's not going to want to make anything to digest. And the enzyme that it needs to make is lactase. So at this point, it doesn't want to be expressing the genes or gene for making lactase. It wants to switch this off. So this is regulation of gene expression. It's going to switch off genes that it doesn't need, and then when it does need it, it would switch it on. So let's imagine it then goes into this environment. Now it does have lactose around, and now it needs to switch on and it needs to express the gene for lactase, because then it can put the lactase outside. It can digest it can digest the lactose, breaking it down into glucose and galactose. And then it can absorb those things and it can use them. So how does that happen? And what are the kind of elements that are involved in that? So here we have the elements involved in regulating the expression of lactase. Up at the top, this is our DNA strand. So regulation of gene expression can work at the level of the DNA, because the DNA will be used to make the mRNA. And so if we can stop that process from happening or make it happen more often, we will either increase or decrease the amount of mRNA we make. And there you can see you've got a region down here on the DNA. We've got this region which is the promoter, and we looked at that in transcription. The promoter is a sequence near, well, it's not just near a gene, it's part of a gene, but it's near to the coding region of the gene. So the promoter was that sequence that allows RNA polymerase to bind. If this gene is going to be expressed, we need our polymerase to come and bind to that sequence and then start moving along. Okay, but we can see here that there's something which is near to the promoter. We've got something else. We've got the RNA polymerase which wants to bind there, but near to it we've got something else. We've got this other sequence here which we can call the operator. And this operator, it could have different functions, but one of them is it could silence. So to silence means to switch off. So here you can see we've got something which is binding to, and so the operator is just it's a sequence of DNA. So just like the promoter was a sequence in the DNA that's recognized, this is a sequence that can be recognized, and it's near to the rest of the gene. So this is the gene for making, this is the information there for making lactase. Okay, above it, what is binding to this operator, this silencer sequence? This is the repressor. So a repressor, you can see, has attached, it can bind to the operator region. And if it's there, it's like a block. It stops the RNA polymerase. It now can no longer bind. So it can't bind, and if it can't bind, there can't be any transcription. And that means overall we wouldn't get transcription. So when would we want this to happen? We would want, in this case, we'd want the repressor to be active if there is no lactose. So when there's no lactose, the repressor comes and binds, it stops the RNA polymerase, it stops the transcription. We don't get any mRNA made, and therefore we don't get, in the end, we're not going to get any lactase. Now, conversely, when we do have, the way the whole repression works is if you have lactose, the lactose itself can bind to the repressor. And when it binds to the repressor, it means the repressor doesn't work. So when we have lactose, we stop the repressor from working. The polymerase can bind. It's bound here. Bound polymerase. It can then run along and it can transcribe. But if we don't have lactose, then it doesn't bind to the repressor. It wouldn't be there. There'd be no lactose there. And therefore, it doesn't stop the repressor. So the repressor would go and bind, and it would then be inhibiting the transcription. And so that's what we had a look at in the first picture. No lactose means our repressor binds, it blocks the RNA polymerase, and we get no lactase made. As soon as we get lactose coming along, it would bind here, which means that this wouldn't be able to bind. We wouldn't have the repressor there, and now polymerase can run along and transcribe, and then we'd get the lactase being produced. So we've got three different major types of DNA sequence. We've just looked at something which was a silencer, a sequence which is going to kind of work to help switch off transcription, where a repressor protein binds to this non-coding region and it blocks or reduces transcription. The other thing that we could have is we could have an enhancer region, where a protein would come along, an activator, it would bind to the enhancer, and then it would be an increase. So this is only one direction, an increase. And then the other kind of DNA sequence that we looked at was the promoter and RNA polymerase binding. So we've got these kind of three different sequences. Promoter for the binding of RNA polymerase, which is always there within genes, and then they may have silencers nearby which work on repressors, or they may have sequences which are enhancers where activators bind to increase the rate of transcription. Our next question is, how can gene expression be changed by environmental factors? So we just saw that really in that example, the idea of lactose being present in the environment changed gene expression in E. coli. If lactose was there, they switched on a gene and they started expressing it, started using it. So what about these bunnies? They're an example of how environmental factors affect gene expression. So they look like they're different species, maybe different types of bunny rabbit, but these are in fact exactly the same type of bunny rabbit. And these black ears, they are caused by cold conditions. As crazy as that sounds, and this bunny was in a warm environment. It was in an environment plus 25 degrees C. And these bunny rabbits here were in colder environments, and their extremities, you might know that if you're in cold conditions, your nose and your ears get cold. The extremities, as they're called, the furthest places, they get cold. And you can see that this has led to a difference in the pigment. And where it's cold, the cold conditions lead to more transcription of the pigment, which makes the ears go darker in colour. And it's called gene C. It controls fur pigmentation. These are Himalayan rabbits. It's active when the environment gets colder, between 15 and 25. So above 25, it's inactive, and we get the white bunny rabbit. Our next fascinating example of the idea of the environment influencing gene expression is this. This is a human embryo. Now you can see at this point it's already started to get hands. But at some point it didn't have hands, not with the normal 5 digits on the hands, and they would have started off without having hands. It would have been a kind of bud. So the question is, how do your fingers know to be your finger? How does it know to be a finger, a forefinger versus a thumb? This picture shows the kind of development of the structures as they begin to grow. And you can see it started off as just this kind of round thing. And the answer is a really surprising one. There is a structure which gives out chemicals, and they diffuse. And these chemicals which influence development are called morphogens. And there are many different ones, and it's still a real area of research in terms of how it works. But if you imagine, if you've got a point source over here, as chemicals diffuse out, there will be some concentration gradients. At the start, the concentration would be high, and as you get further away, the concentration would drop. And in essence, the concentrations of these morphogens, these different morphogens, will tell these different digits to grow in a particular way, to become the thumb. What this might look like in a simple model is that if you have a source of a morphogen, and so in this case, let's take it as the other way around. If this is the source, where we have, you can see A, that would be our high concentration, down to D, which would be our low concentration, because we'd have lots of whatever, let's do it as red, lots of the morphogen here, and you'd get less in this region, and then not very much at all there. And in fact, you've even got so you can see it's a gradient. And what's amazing is that cells have a way of taking that gradient and using it to kind of quantify. They can quantify how much of the different types of morphogen there are, and then that will lead to transcription of different genes, causing, in the end, different cell types. Going back to our example here with the human hand, it would mean that if the source was over here, then there would be a low concentration there because it would be further away, and there would be a higher concentration here. And these various gradients of different morphogens lead to tissues knowing how to develop in embryonic development. And here's another example in humans. So what do we see here? We see environmental conditions which are affecting gene expression. And what's the different environmental condition? Well, this is to do with UV exposure. Here, this person has had UVA and UVB sunlight striking his skin, and as a result, the cells in the skin, they start to produce a pigment, and that pigment, it leads to the transcription of melanin. Melanin is the pigment which makes you darker skinned. So the environmental condition of sunlight exposure has caused melanin to be transcribed and has caused the suntan. So that's another way that gene expression can be regulated. So here's our summary. You can have regulation of gene expression in different environmental conditions, e.g. the production of melanin in UV light. We looked at embryonic development where morphogens are produced, and the concentration of those morphogens reaching the cell determines how the genes are expressed and even what type of cell it becomes, leading to different tissues being formed depending on the position in the embryo. We also had the example of temperature, coat colour, and you could either talk about cats, where we've got one, or you could talk about the Himalayan rabbit, where higher temperatures we saw led to the production of dark patches. In cats, we've got coat colour normal is C, which leads to a dark coat colour, and you can get a mutant one, little c, which only leads to normal colour at below body temperature. And so that in cats it will give different colours as well. The example that we looked at was the Himalayan rabbit, and in the rabbit, if conditions were cold, it would lead to the pigment being produced. And if they were warm, it would then, you get no pigment, so they would be white in colour, totally white. So yeah, this is expression due to the environment, and that example in cats is in Siamese cats. The final thing that we'll look at is the idea of regulation at the level of nucleosomes and epigenetics. So how do nucleosomes regulate transcription? If you remember, nucleosomes were the histone proteins, the kind of 8 histone proteins around which we wrap DNA. So how can they regulate transcription? The first thing is that if the cell is copying and we have supercoiled DNA, and everything's very tightly coiled up, then we don't get any transcription. So if things are very tightly wound, we don't get any transcription. And so this is a key principle. If the DNA is more loosely associated with nucleosomes, we might get increased transcription. So what makes the DNA, if the DNA is here, what makes the DNA associate with the histone protein at all? Well, they must have an attraction. They associate because there's an attraction. What charge does DNA have? Well, DNA has the sugar phosphate backbone, and the phosphate is negatively charged. So DNA overall has this negative charge. So what would it then attract to on the histones? Well, it would attract to positive charges on the histone proteins to associate. So in histone proteins, there is an amino acid called lysine. And lysine has a positive charge. And that will cause the DNA to kind of tightly wind around it. Now, if the DNA is very tightly wound, this leads to less transcription because the polymerase is less able to access that sequence. If it's loosely associated, that would tend to lead to more of the transcription happening. Okay, so we can then think about how can we regulate that attraction. And one of the things that happens is acetylation. An acetyl group, which for the chemists is C2H3O, it has a negative charge to it because of the O. We often have, so we saw that with water, this idea of negativity. So acetylation can happen. We can add an acetyl group on, and that, if we're adding an acetyl group on to the histones, so it's acetylation of the histones, the proteins that the DNA winds around, if we then are adding in a negative charge, it's going to cancel out. And so we won't have this positive. So if we think back to this diagram here, if overall we had positive charges attracting to the negative DNA, if we now go in there and get rid of that, if we get rid of the positive charge, now we don't have anything for the negative to attract to. So acetylation, if it's getting rid of that attraction, there's less attraction, which means that the DNA is going to be more loosely associated, which means we would get more transcription. So here we see acetylation adding acetyl groups. This would lead to increased transcription because it's cancelling out those positive charges, making the DNA more loosely associated. So you can see here it's got the idea of the DNA kind of being more stretched out there, not as tightly wound up. The other thing that can happen is we can get methylation, and this is methylation of DNA. You can see here that the cytosine, or the C's here, have had a methyl group, which is CH3, added onto them. And methylation tends to be associated with a decrease in transcription. So by making these changes to DNA and making the change to histone proteins, we can, for methylation overall, it tends to be a decrease in transcription, and acetylation tends to increase transcription. Here's our summary. So nucleosomes are made out of histone proteins. They are involved in the packaging of DNA. There can be the chemical modification of histones which influences whether the gene will be expressed, whether it will be transcribed. So the amino acid lysine is normally positively charged, and that means that it interacts with a negatively charged DNA. And that would form a condensed, tightly packaged structure which would inhibit transcription. But if you acetylate the lysine, that would remove the positive charge, causing it to be less condensed, looser, and allowing more transcription to take place. Our final one is, what is epigenetics and how does it influence gene expression? Well, epigenetics is the study of chemical modifications So the other thing that we know definitely happens is mutations of DNA. But this is not epigenetics. Mutations are definitely passed on to the next generation if they're in the sex cells, the sperm and the egg. Whereas epigenetic changes, these chemical modifications, they actually tend not to be passed on. So when we're talking about epigenetic changes, we mean above the level of changing the DNA. It's not changing the DNA sequence. But things like acetylation of histones, which increase transcription rate, methylation of, this is incorrect, methylation of DNA. I will change that. The methylation of DNA, which overall tends to reduce the transcription rate. Switching off genes, causing DNA to become silenced and more condensed rather than open and transcribed. So the other, with acetylation, it was causing the DNA to be more open, more transcribed. Now the idea of epigenetics is that now scientists have found that some of these changes are passed on. Previously it was thought that all of those changes were wiped clean. When you made sperm and egg cells, it would wipe clean all of these changes. And that's true of almost all the changes, but we think now, studies suggest it could be about 1% of methylation and acetylation could be passed on to the next generation. For instance, an example that they found is that if you have diabetes as a mother, it predisposes the child to diabetes, and that is an environmental change through methylation and acetylation, through epigenetic changes. And so the example was given is that a traumatic experience that your grandmother had could in some way be passed on through epigenetic changes, and they might be present in you, even though they might be a very small amount.